we're going to begin talking about some of the gram-positive rods. And then as I was redoing my slides from the last time I taught this class, and I realized I was presenting it today, I was like, cool, I'm talking about anthrax on September 11th. That was a good plan. Good plan, Van Laar. So we're talking about anthrax today. All right, so the first genus that we'll talk about in the gram-positive rods is the genus Bacillus. So remember, the shape of a rod is also called the Bacillus shape. So something can be a Bacillus shape and not be in the Bacillus genus, but every Bacillus genus member is a Bacillus shape. Sorry, it's confusing. We mostly will call them rod shape, but remember the technical term for a rod shape is Bacillus. And it's named that, and the genus, right, is named that because that's what it means, Bacillus, small rod. The two major species we talk about as potential pathogens are Bacillus anthracis, which is the cause of anthrax, and Bacillus cereus, which is sometimes more commonly associated with foodborne illness. So anthrax means charcoal, and what happens is when patients get anthrax in the skin, the cutaneous form of anthrax, it forms a black wound. So that's why it's anthrax. Now, Bacillus anthracis by itself is actually not that big of a deal. What happens is that the bacterium contains a plasmid called PX01 or PX01. So remember, a plasmid is an extra chromosomal piece of DNA. It replicates on its own. Okay. Sometimes they can be shared, but in general, it's just this piece of DNA that replicates on its own. And so every time the bacterium replicates, it should replicate the plasmid as well. There are 3 genes on this plasmid that are important for symptom development. The 3 genes are lethal factor, protective antigen, and edema factor. So lethal factor, protective antigen, and edema factor. Protective antigen is a component of both toxins produced by anthrax. So protective antigen is part of both toxins, but if lethal factor is combined with protective antigen, you get the lethal toxin. And as you might imagine, that causes cell death. It's lethal. If you combine protective antigen with the edema factor, you get edema, right? Fluid retention. So it makes a toxin called lethal toxin and one called edema toxin. That production of these 2 toxins is really how Bacillus anthracis causes disease. There's a soil bacterium that is used in genetic biotechnology called Bacillus thuringiensis. Chromosomally, they're identical. It's the presence of the plasmids that separates the 2 species. So these plasmids are super important for anthrax. There is a second plasmid called PX02, which encodes genes to produce a capsule. Now this is challenging to study, this capsule, because the bacterium, as to this point, will not make the capsule in vitro. So there is something about the in vivo condition where the bacterium will activate the plasmid and produce the capsule. And remember, a capsule would always help with immune evasion, adherence to host cells. So what we worry about is the toxin. And what they're showing you here, right, are the gram-positive rods mixed in with other cells. Now, this is not a brand new infection. Bacillus anthracis has been around forever. And it's normally a disease of herbivores like cattle. In the US, there were only 5 cases of anthrax reported in people between 1981 and 1999. Very, very uncommon. There have been 2 major releases of anthrax. One was accidental in the Soviet Union in 1979. One was purposeful as a type of bioterrorism in the US in 2001. So again, it is a disease of herbivores, and I told you it's been around forever and ever and ever. One thing that maybe you remember about the genus Bacillus from when you took bacteriology is that it is one of the few genera that we study that forms an endospore. Remember, you had to do the malachite green stain, you had to boil it for like 5 minutes. Those endospores are very, very resistant structures, right? The only way to get rid of them is by using the autoclave or other sterilizing techniques. In fact, we use Bacillus subtilis often as one of the organisms to test that the autoclave is still working. Can we get rid of all the spores? Well, these spores can remain viable in the environment for an indefinite period of time. And so this headline, anthrax sickens 13 in Western Siberia, and a thawed-out reindeer corpse may be to blame. Well, reindeer are herbivores. Reindeer are susceptible to anthrax. And so there are, apparently in Siberia, reindeer scattered throughout the permafrost, right? Reindeer corpses. And with climate change, as the global temperatures rise, in some of these areas, the permafrost begins to thaw. The spores can become aerosolized and be inhaled by people who come by. So this also means that it could be dangerous to live in an area where previously cattle had been raised, because if cattle died of anthrax, one of the things they would do is bury the corpses. So it's fine to bury the corpse, but those spores could still be viable in the soil for who knows how long. When we talk about anthrax, there are 3 forms of disease, and I alluded to one already when I talked about how it got its name because of the dark black wound. So there is a cutaneous form of anthrax. So it can be at the very beginning a painless like lesion or like infection that will eventually ulcerate and lead to eschar formation. Not eschar is going to be the dark black wound. Painful lymph nodes, edema, so swelling, and systemic signs may develop. Now with a disease like anthrax, just because you see systemic signs does not mean that the bacterium itself has gone systemic. The bacterium could remain localized in that wound, but because it's producing the toxin can travel throughout the body and encounter the different tissues. So just the presence of the toxin in the body is what's causing the symptoms. There can also be gastrointestinal anthrax. You ate something contaminated with spores. Wherever the spores germinated, you'll get ulcer formation, mouth, esophagus, stomach, intestines, and there you'll get the regional lymph nodes swelling, and you eventually can get sepsis because they can break out of those very thin tissues and get into the rest of the body. Again, you also could have them stay localized in the intestine, and you could have those systemic symptoms appear because of toxin disseminating through the blood. What people tend to think about, though, when they hear about anthrax is inhalation anthrax. And this is when a patient has inhaled spores. The spores then will germinate in the lungs, and it's a very initially non-specific disease manifestation. It looks like a respiratory tract infection. There might be mucus production, difficulty breathing, coughing, but eventually, and sometimes relatively quickly, as the toxin is being produced, you get the fluid coming into the lungs, you get swelling of the local lymph nodes. Patients can develop symptoms of like meningitis, and most patients with inhalation anthrax will die unless treatment is begun immediately. And treatment has to be basically a combination of toxin neutralization, respiratory support, and antibiotics to get rid of the bacteria. So you have to stop the toxin from acting. You have to keep the patient alive from with whatever tissues have been damaged, and you have to get rid of the bacteria so they stop producing more toxin. Okay, so again, I told you that this I thought was I'm like, what a weird coincidence that I'm presenting anthrax on September 11th, because I imagine most of you, if you were alive, were not very old. You were probably very small children, or babies. You're also very cute, small little babies. I was also not quite an adult, but I was very close to one. I was 17. I was getting ready to go to college, and it was September 11th, and I think we all know what happened that day. In October, so like a few weeks later, the first victim died on October 5th of 2001. So again, you were probably very young, maybe not super in tune with like the political climate and how everybody was feeling. Well, everybody was really freaked out. And then when people started dying of anthrax, everybody got even more freaked out. And so what happened was, they call it Amerithrax because everything needs a name, right? So it's American anthrax. Spores of Bacillus anthracis were sent through the U.S. Postal Service, so through the mail. And letters containing spores, of course, went through a number of postal facilities, were sent to media outlets, and were also sent to a congressional office building in Washington, D.C. So 5 victims died of inhalation anthrax. A couple of them were postal workers. Another was an elderly woman from Connecticut, a Manhattan hospital worker, and an employee at a Florida tabloid magazine. And it may have just been through cross-contamination, because these were, you can look up the pictures of what these letters looked like on the internet, but one of the Senate Majority Leader at the time was a guy called Tom Daschle, and there's like a letter and it says, I don't know, something like 4th grade class, whatever, like the return address. It's all in block letters, and it looks like maybe somebody with poor handwriting or like a child might have written it. And then it says, to Senate Leader or Senator Tom Daschle, it gives his congressional address, and it's just a thing full of spores. So as these were transiting through the mail, it was very likely they came into contact with other pieces of mail. And so that's how they think especially the hospital worker and the elderly woman in Connecticut probably got exposed. So it was very scary that all these people were getting anthrax, there were spores, they had to close down the congressional offices and close down all these postal facilities and like bring in super sterilization to get rid of all these spores. And they never made an arrest. They interviewed, you can see here, 10,000 people, more than 6,000 subpoenas. The Postal Service bought millions of masks and gloves. 300 postal facilities were tested for anthrax. 32,000 people were given prophylactic antibiotics. It was a really, really big deal. And so for a long time, nobody knew what happened. There was one guy they thought had done it. They investigated him. It turned out he hadn't done it. He sued the government for millions of dollars. He won. And then in late July of 2008, a former Army bioweapons researcher died by suicide. And it turns out that basically it was him who had done it. So it was a U.S. citizen. It was a guy who worked at the Army Bioweapons Laboratory at Fort Detrick, and nobody really knows why he did it. Mental health issues, I guess mental health records suggested he should not have been cleared to work there, but he was, so that was it. It wasn't some, scary boogeyman from another country. It was somebody from right within.
So this anthrax happened after September 11th?
Yes. It happened about 2.5, 3 weeks after.
That's
Yeah, so everybody was like, I mean, we were freaking out. You had no idea what was going on. You're like, okay, who's trying to kill us now? Like, what's going on? Is it safe to check the mail? Like,
So
people
that
were.
happened because of that attack?
That's probably what kind of gave him the idea to send it, was that attack. And then it wouldn't be blamed on him. It would be blamed on the terrorists, right? It was Al-Qaeda. Al-Qaeda did it. So that's how he escaped being detected for a long time. But when they genetically tested the strain of anthrax, it matched the one that they work with in the lab in the Army. Yeah, it was immediately following. So it was a weird time. But we still do see anthrax in cattle. It is not rare. So anthrax has been confirmed in cows in North Dakota, Southwest Texas. Cattle still get anthrax. And so even though the risk is low for the ranchers and the other people who work with the cattle to get anthrax, it is possible. Most commonly, though, what you would see in people who work with infected cattle or the ranchers is it would probably be the cutaneous form. Like if they were slaughtering the cattle and they had a wound, some spores got in, they could get that cutaneous form. The inhalation form is only caused by exposure to spores. It has to be the spores. And they used to call it wool sorter's disease because people who worked with hides, especially goat hides, as they had been dried out, could get it. Of course, we've seen it in the US. It was only the second time that a biological agent was used as a type of terrorism in the US. It was the first time that killed people. It has a very low infectious dose. You don't have to inhale a ton of spores in order to get sick. And this is going to sound really creepy when I say it, so I'm sorry that it sounds creepy, but it's not a great bioweapon. There are better, which is the part that sounds creepy, that they're better bioweapons. It's not a great one because it doesn't spread from person to person. So if a person has inhalation anthrax, it is almost zero chance that you will get inhalation anthrax being exposed to that person. So in terms of if you are trying to wipe out the population, not great, because you just, stop opening the mail. If you're trying to scare the crap out of a whole mess of people, which is mostly the goal of being a terrorist, you want to scare people so they'll do what you want, that's pretty effective. It scares people. So with the cutaneous form, again, you're going to see a papule at the site of infection. You can see like how the ulceration and the tissue necrosis can spread from the initial wound. It can go systemic, but often doesn't. And generally, even if left untreated, the mortality rate is relatively low, about 20%. So in patients who are treated, it's even lower. Gastrointestinal is going to be one of the least common, but it also can be 100% fatal if left untreated. You'll get the ulcers that form in the mouth, the esophagus. If the spores germinate in the intestines, it can be accompanied by nausea and vomiting. And again, that mortality rate is really high. With inhalation anthrax, patients can be asymptomatic for 2 or more months. You're waiting for the spores to germinate, for the numbers of cells to get high enough to produce enough toxin to cause the symptoms that we would see. And again, a lot of those symptoms that are seen are common, like upper respiratory tract infection symptoms. But it's not pneumonia, because pneumonia is going to be huge pockets of infection in the lungs, whereas this could be a very small infection just producing a boatload of toxin. So it's going to be more fluid buildup in the lungs than infection buildup. The spores can be phagocytized by the alveolar macrophages and transported to those local lymph nodes, and that's where you might get the lymph node swelling and pain. So again, it's the release of toxins that causes disease. So it's not actually pneumonia. Pneumonia means like it's the infection causing most of the symptoms. It's actually the toxins in this case. And if, again, left untreated, but even if treated, it can be very difficult to to help your patient because it progresses so rapidly. Patients can go into shock and die within about 3 days. So then what do we do for diagnosis and treatment? You could do a gram stain of the blood. Gram-positive bacillus-shaped organism in the blood. PCR, you could look for the genetic material. Antibiotics generally work pretty well. We don't see tons of resistance in Bacillus anthracis. Ciprofloxacin or doxycycline would be the drugs of choice, though you can combine it with other antibiotics to really make sure you hit it and clear it before too much toxin is produced. To help kind of reduce the spread of infection, it is recommended that if you have cattle that they are vaccinated. So there is a vaccine for anthrax. It should be given to cattle. It is sometimes given to people under very special circumstances. So people who work in high-risk professions can be vaccinated. This is mostly going to be the military, people who work in labs that work with this organism. But you're not going to, unfortunately, be able to walk into CVS and get an anthrax vaccine. Okay. So questions about anthrax? Yes.
So since I'm searching right now, it was a letter that was sent to the post office.
There were lots of letters that were sent.
So was it something they put on the letter that they got infected, or I don't know?
It's just literally spores. So you grow up a culture of the bacteria, you dry it out, and it's got a lot of spores in there. It's a powder. You put it in the mail. Yeah. Yeah, because it forms the spores, it gets dried out. You don't have to do anything special with it. You don't have to keep moisture in there. You don't have to protect it. It can go through the mail. It can get too hot. It can get too cold. It can get irradiated. No problem. Yeah. Yeah, and there were a number of letters that were sent out, but the one I'm picturing in my head that they showed on the news constantly was the one that went to Tom Daschle. And so they had to shut down that whole wing of the congressional office for, I don't remember exactly how long they had it shut down, but it was for a long time. Any other questions?
Did they find out who wrote the letter?
The guy who killed himself is who they think it was.
Okay.
No. It was a scientist who worked at the Army base. Yeah. So it was an American citizen who had mental health issues, probably shouldn't have had the clearance to work at the facility in the first place, but a lot of things were different, Mental health screening was different. I was reading a thing on Reddit where they were like, what's the least serious thing that you've seen change since September 11th? And I remember I had just come back. I graduated from high school and I went to Hawaii with my friends. And we came back, I saved up $800, which was big money to me from working at the hometown buffet. Seriously, $5.75 an hour. It was awful. And we came back and our parents could meet us right at the gate, right? Your parents could walk right up to the gate. They could drop you off. You could wait for your friends. When they got off the plane, you can give somebody a hug, and there was no security like that. It was crazy. And now the first time I flew after that, I was like, I got to take off all my clothes. I can't bring anything on the plane. Where are my snacks? So that was probably the least, that sucks. You have to get to the airport 3 hours early. You used to just, got to park and walk in. That was it. It was so easy. It was nice to fly before then. I hate flying. It's the worst. Any other questions about anthrax? Okay. The other Bacillus species that is relatively common in causing human disease is Bacillus cereus. And generally we see Bacillus cereus as a gastrointestinal infection. So it also produces toxins. These toxins, though, are enterotoxins. They're gastrointestinal toxins. There is a heat-stable toxin. So that means no matter how much you reheat your food, if the toxin is already on your food, you will get sick. The heat of the microwave won't kill or destroy the toxin. So you're not even necessarily ingesting the organism in that case. You're just ingesting the toxin itself, and it causes emetic form of disease or vomiting. It's often associated with contaminated rice for whatever reason. Rice tends to be one of those that, like, you'll cook it, you'll leave it out for a while, maybe then you put it in your fridge, and then you're like, oh, I'm gonna have some more of my rice, and you like heat it up in the microwave, and between usually 2 to 6 hours after consumption, you'll have vomiting, nausea, abdominal cramps, and it probably will only last a few more hours after that, 8 to 10 hours or so. But again, the toxin is heat stable, so once it gets into the intestines, it can start working immediately. That's why the symptoms are relatively rapid onset. But with that vomiting, right, you're moving the toxin back out of your body, so the symptoms tend to go away relatively quickly. The heat labile toxin, the one that would be destroyed by heating, is produced oftentimes when you've eaten something contaminated with the bacterium. This is more of like an infection. So you've got the bacterium maybe in your gut. It's producing this toxin. Generally, you're looking at maybe 9 hours or so after you ate, and it will cause diarrhea more commonly than it would vomiting. So it's mostly a foodborne infection. I couldn't even tell you how many people maybe have had that. How many times have you eaten something and you either threw up for a few hours or maybe you had diarrhea for a day, but you don't go to the doctor to get diagnosed. You're like, well, I'm not gonna eat that again. And then you eat it again later and it's fine. Like once you get over it. Yeah, my thing was I ate a frozen burrito once that made me throw up. And I couldn't eat the burrito again for like 3 years. And then finally I had one. It was totally good. So I don't know. Right? But I don't know what it was. Was it? No, you know what? It was. I love Taco Bell too. Taco Bell's the same thing.
I had it once when I was 8, threw up. I haven't had it since.
Oh, see, yeah.
Nope.
But I bet you if you go, you'd be like, man, it's good.
Even the smell of it now.
Oh, yeah. Oh, those frozen burritos for like I said, 3 years. I'm like, stop cooking these dang burritos around me. They're gross. I got over it. But you don't know. The point is, you eat something, you throw up, you almost can't unless it's,
I have a metaphobia though, so that might.
That'll do it.
That'll do it.
But, it's like, but you don't know what caused it. Like, you're like, I ate Taco Bell and it made me sick, but I don't know what was on the Taco Bell that made me sick. I don't know what was on the burrito. So I imagine that people get these particular infections, things like Bacillus, cereus, and some of the other foodborne illnesses we'll talk about, way more often than we think, because again, if you throw up for 10 hours, you're not going to go to the doctor, especially when you stop throwing up. You probably can't even get an appointment for the next 3 weeks. You're not going to go to urgent care because you had diarrhea for a day. By the time you I don't know, I wouldn't. Because by the time I felt better, I'm like, yeah, actually, why would I go to urgent care now?
Working at a facility, I can confirm that they will go to
Oh.
the doctor if they've been having diarrhea for 3 hours.
Oh no, I'm like, no, I'm gonna wait this one out. Like the vomiting, I'll wait it out. If it's been a few days, then I'll okay, it's been a few days, I don't feel well, you get dehydrated, but otherwise, maybe I just don't like going to the doctor. I'm like, I'm not paying the urgent care upcharge. I want my $15 copay. So it's hard to diagnose what exactly is causing foodborne illness, but oftentimes if we do know something that's causing a specific foodborne illness, it's because it came from a restaurant or a localized source where a lot of people got sick. So when we talk about things like norovirus, it was a few years ago, before I moved back to California, but Chipotle restaurants, there were a bunch of Chipotle restaurants all around the country that had norovirus. And one of them was this one right here. I still go there. But, these foodborne illnesses can go around. The more serious form of disease caused by Bacillus cereus is if it gets in your eye. So sometimes it can be associated with ocular disease. And while the gastrointestinal infection is more common and of course much more mild than the ocular disease, we can see ocular disease, and there are a few different toxins that can mediate the disease. You have a necrotic toxin, which causes tissue death, right? Cell death. You have a cereolysin. Anytime it says lysin, it can lyse different host cells. And a phospholipase C, which also can break down the phospholipid bilayer of the cell. So it basically produces 3 toxins that are trying to break down your cells in different ways. And this can lead to rapid destruction of the eye. These bacteria can replicate relatively quickly. These are some of our faster bacteria. So once they get in and they're producing this toxin, it can move pretty quickly. In patients who are immune compromised, if the bacterium gets into the lungs, it can cause pneumonia that mimics anthrax, but it causes infection in the lungs. So the symptoms can be similar. You would see patients who have spore-forming gram-positive rods, so you might think anthrax, but it doesn't produce those same lethal and edema toxins. So it is an actual pneumonia. It's an infection, right? It's not edema toxin or lethal toxin causing the symptoms. And again, this is really what we see only in immune-compromised patients. Okay, I thought this headline was kind of interesting. Ed Yong is a really good science writer, so he writes about science for popular magazines, tries to make it a little bit more accessible for people. So a strange type of anthrax is killing chimpanzees. It was actually a new strain of Bacillus cereus that produces some of the same toxin that anthrax can produce. Remember, these toxins are on plasmids. The toxin genes are on plasmids. Bacteria love to share their plasmids. So Bacillus cereus has some of the same toxin genes. They don't know exactly how, like when that happened, where they got them, but it's likely to wipe out a local chimpanzee population within the next 150 years or so. So it's just interesting, I thought, to think about. Bacillus cereus is found in the soil. Most Bacillus species are. Even Bacillus anthracis, found in the soil. It can be cultured from individuals or from food that has been associated with causing illness. And as you would expect, as with most gastrointestinal infections, the infection is self-limiting. The patient will feel better and clear whatever's causing either the toxin or the bacterium itself without medical intervention. That's what we mean by self-limiting. It will go away without medical intervention. The ocular infections must be treated promptly. We don't see a ton of resistance, so you have a handful of drugs that are good. Things like ciprofloxacin, vancomycin. And that's that. Questions about Bacillus cereus or Bacillus anthracis before we move on to Listeria and diphtheria. Okay. Perfect. Okay. So this set of slides, and we'll finish diphtheria on Wednesday, are about 2 other gram-positive rod-shaped bacterium that are relatively common causes of infection in people. So the first is the genus Listeria. The genus gets its name from Joseph Lister, so of Listerine fame, Listerine, Listeria. So he was a scientist who studied bacteria. They are non-spore forming, so that's one way that you could differentiate them from something like Bacillus. They're all gram-positive, but Bacillus forms spores, Listeria does not. They are uniform in shape, and that is what helps to distinguish them from Corynebacterium, which is the genus we'll talk about after this. And what I mean by that, and what we'll see when we look at images of Corynebacterium, is that they're all rod-shaped, but they can be like longer or shorter, like Corynebacterium. the rods don't look consistent on the slide. There could be like different size rods, maybe some are a little wider. They call it pleomorphic. So they're all rods, but they're all slightly different. With Listeria, they're all the same size rod. So that's what we mean when we say uniform in shape. They're relatively short. You might remember looking at something like Bacillus subtilis or even Bacillus megaterum, right? Giant rods under the scope. These are usually pretty short. One of the things that makes Listeria, in my mind, so dangerous is the fact that it grows very well at a very wide range of temperature, anywhere from 1 to 45 degrees. It grows equally well, basically, in the refrigerator as it would in your body. And that's scary because we tend to think of the refrigerator as the place where we keep our food so bacteria doesn't grow on it. And Listeria is like, I got this. I will grow in here. It can be beta hemolytic. It's not as strong as some of the things like staph or strep that we've talked about before. So you may not see a huge clearance, but you will see a clearance around the colonies. And we typically think of Listeria monocytogenes infection as a foodborne illness. So not respiratory, not wound, you ate something or drank something. So one of the cool things that Listeria does that helps it to evade the immune system really, really well is it actually wants to be phagocytized. So this is one of those bacteria that when it encounters phagocytic cells says, yes, please. Or if it comes in contact with epithelial cells, it wants to get inside of host cells. And then it contains a couple of different enzymes, listeriolysin, right? Any lysin, O, and a phospholipase C, that help it break apart the phagolysosome. So remember, during phagocytosis, when an organism is phagocytized by one of our cells, it forms a vesicle around the cell. We call that the phagosome. And then there's a lysosome within the cell that contains antimicrobial compounds. Acidic pH, reactive oxygen species, maybe lysozyme, whatever, right? Those two fuse, so the idea would be that the lysosome merges with the vesicle containing the cell, dumps all of those compounds, changes the pH to make it more acidic to kill the bacteria that's in there. What Listeria monocytogenes will do is basically digest the phagolysosome and escape. So it gets out, and now it's inside the cytoplasm or the cytosol of the cell, and that's where it will replicate. So one, it does immune evasion by escaping the phagolysosome. Once it's inside the cell, in the cytosol, it's escaped the phagolysosome. The other way that it evades the immune system is it doesn't exit the host cell to go infect new cells. It doesn't come out, it doesn't cause the host cell to burst. It literally rearranges the actin of the host cytoskeleton and pushes its way into the neighboring cell. So it's like if you live in an apartment complex and you want to go into your neighbor's apartment, you normally would walk out your front door, knock on their door, and they would let you in. And, you could be seen by people when you do that. So you don't want to be wearing your gross clothes because somebody might see you. Or you could do like Listeria and just bust through the wall into your neighbor's apartment. Your neighbor probably wouldn't like that, but at least you could wear your scrubby clothes and nobody will judge you. Your neighbor might because you broke into their apartment, but you just pass through the wall. And you pass through the wall and pass through the wall, and nobody's going to see you do that. And so that's what Listeria does. It pushes into neighboring cells, which again hides it from the immune system. Antibodies can't find it. Phagocytic cells can't find it. Most T cells can't find it. It hides, so it can spread. And so it has proteins regulated by a particular gene, and I won't make you remember which particular gene, that allow it to do that. And so this is kind of what happens. So this poor guy right here is about to get Listeria, that gene that regulates all of these other genes turns on, and so the bacterium is phagocytized. It has proteins to allow it to attach to the host cell, so it wants to bind the host cell. Binds the host cell, gets phagocytized. It uses its phospholipase C and listeriolysin O to lyse the phagolysosome, and then it has a protein called ACT-A, which it uses to rearrange the actin, and it literally pushes into the next cell. And what they're trying to illustrate here is that the original cell's membrane is still covering it, so it never is exposed to the external environment. It constantly is covered by the host when it gets into the next cell. And it can do that over and over and over again. So that's how it evades the immune system. The bacterium can be found from both environmental and fecal sources. Most people get the infection from contaminated food. It can be dangerous for a pregnant woman because the bacterium can cross the placenta and be transferred from the mother to the fetus. Not a lot of infections, especially bacterial infections, can do that, but this one can. It is not a terribly common foodborne illness. We tend to think of things like E. coli, Salmonella, Shigella, right? More common bacterial infections. But it has one of the highest mortality rates. So when patients do get this, more of them tend to die. So here's a case of Listeria in a patient who's immune compromised. He was a 73-year-old man with rheumatoid arthritis, brought by his family to the hospital because he had decreased consciousness, 3-day history of headache, nausea, and vomiting. His current medications, so he was taking a monoclonal antibody, methotrexate and prednisone, to basically reduce the inflammation and reduce the swelling from his rheumatoid arthritis. So some of those steroids that cause the reduction in inflammation, they cause the reduction in inflammation because they decrease the immune response. So patients who are taking these drugs can technically be considered immune compromised. Their immune response is lowered to prevent the arthritis symptoms. On physical exam, the patient had a stiff neck and a fever. So stiff neck and a fever, generally indicative of meningitis. So the stiff neck symptom is pretty classic. He had a slightly elevated heart rate and an elevated blood pressure because meningitis was suspected. Stiff neck, right? And then headache, nausea, vomiting, decreased consciousness. They tested blood and cerebrospinal fluid. Gram stain of the CSF was negative, but Listeria grew from the blood and CSF. So just because you don't see the organism in the CSF, it could just be at too low of a concentration for you to actually see it, right? Because when you're looking at the microscope, right, you probably remember this from if you did your mixed cultures in the lab, if you didn't grab enough, you didn't really see it on the slide. If it's not at a really high density within the CSF, maybe you don't see it, so you culture at the same time. The patient was treated with vancomycin. They discontinued at least one of his rheumatoid arthritis therapies, and he made an uneventful recovery despite using a less than optimal antimicrobial therapy. This monoclonal antibody has been associated with dose-dependent monocytopedia. It reduces the number of monocytes, which are right where we get macrophages, the monocyte macrophage lineage. And you really have to have an effective phagocytic response that can actually kill those cells to get rid of the bacterium. So he was specifically at risk for this infection. Failure to detect Listeria by gram stain in the CSF is typical of this disease because the bacteria fail to multiply to detectable levels. So again, when a patient is experiencing symptoms of meningitis, even if the CSF looks clear by microscopy, be aware that meningitis caused by a bacterium could still be a possibility. It will grow on most media. It's not terribly picky. We haven't really talked about a picky bacterium yet. We'll get there. We'll talk about some picky ones. But you can do cold enrichment. So let's say you suspect a person has a GI infection with Listeria and you want to plate a stool sample. Well, there's lots of stuff that's going to grow, but most of it only grows at human body temperature. So if you incubate the plate at a cooler temperature, that should suppress the growth of the other organisms and allow for the growth of Listeria, because it will grow at that wide range of 1 to 45. It is motile, which is not common in gram-positive rods. So you could do some type of motility test, and it moves. They will do whole genome sequencing during suspected outbreaks to track where it came from. It can be treated with gentamicin and penicillin or ampicillin. So they'll do a combination therapy. Gentamicin is, I believe, a protein synthesis inhibitor. And then, of course, penicillin and ampicillin are both cell wall inhibitors. So you do a combination therapy to really get rid of it. Sometimes drugs like penicillin, some of the cell wall inhibitors like vancomycin, they aren't great because they don't do well at getting into host cells. And remember, this is an intracellular bacterium for a lot of its life cycle and infection cycle. Gentamicin will get into the host cell. So you want to have a drug that'll get into the host cell as well. To avoid disease, avoid raw or undercooked dairy and other animal foods. Thoroughly wash all produce. I was at Sprouts once in Fresno, and they had a thing that was like, oh, raw milk, next best thing to mother's milk. And I'm like, are you suggesting that people should give an infant raw milk? And yes, they were. And by raw, I mean unpasteurized. I'm like, you trying to kill children? And so we do see outbreaks of Listeria. I think there's one going on right now, and I can't remember what food it's associated with. But protein bars, cantaloupe was a big one because the bacterium was on the outside of the cantaloupe. And if you think about what you do with cantaloupe, or at least what I do, you get a cantaloupe, you cut into it, and then you put like saran wrap and put it back in the fridge. The bacteria are all on the outside. You've just now brought them onto the inside, and they actually grow and replicate in the fridge on your cantaloupe. So now, wash my cantaloupe before I cut it open. Because you think, oh, the thick rind, the fruit's going to be protected. And then this was the one that made me really sad, ice cream. This killed people in a hospital in Kansas because it was actually like replicating and growing in the ice cream that they were feeding to hospital patients. So it's a dangerous one. We'll leave there. And we'll pick up on Wednesday with diphtheria. And again, make sure if you do want to redo